Welcome back once again, my dear students. Today our topic is active transport. And before discussing active transport, let's recapitulate what we have studied about the diffusion process, because diffusion process is a passive process. Okay. In diffusion, there is movement of molecules from higher concentration to lower concentration. Okay. This is the higher concentration side and this is lower concentration side, and the molecules move from higher concentration to lower concentration. That means the molecules are moving down their concentration gradient, and it is not an active process. It does not consume energy because the molecules are moving down their concentration gradient. And furthermore, diffusion is of two types. One is simple diffusion and another one is facilitated diffusion. In simple diffusion, the molecules move directly through the lipids of the plasma membrane, whereas in case of facilitated diffusion, the molecules move from higher concentration side to lower concentration side with the assistance of transmembrane proteins. Okay, so you can say that both these processes of simple diffusion and facilitated diffusion, they are passive processes. Now let us focus on active transport, that type of transport process in which the molecules are moving from lower concentration to higher concentration, and in this process, since the molecules are moving from lower concentration to higher concentration, or you can say the molecules are moving uphill, this is only possible when there is consumption of energy. Okay, when there is consumption of energy, only then molecules can be able to move from lower concentration side to higher concentration side. This type of transport process in which the molecules move from lower concentration to higher concentration, and this process is accompanied by expenditure of energy, it is known as active transport. And depending upon whether the energy consumption is direct or it is indirect, active transport has further been divided into two types: primary active transport and secondary active transport. Now, what is primary active transport? In primary active transport, the molecules move from lower concentration side to higher concentration side. This is the higher concentration side. Okay, there is higher concentration of molecules on the inner side of the cell and lower concentration on the outer side of the cell. Okay, the molecules are moving from lower concentration to higher concentration due to direct consumption of energy. When there is direct consumption of energy, say for example in the form of ATP hydrolysis, ATP breaks to form ADP plus inorganic phosphate, and the energy which is released during this ATP hydrolysis, it is used to drive the movement of molecules from lower concentration to higher concentration. This is known as primary active transport. Now, what is secondary active transport? That type of active transport in which the energy which is stored in electrochemical potential gradients is used to drive the movement of molecules from lower concentration to higher concentration. That type of active transport is known as secondary active transport. Say for example, this is an H positive ATPase pump. The function of this H positive ATPase pump is it moves H positive ions from inner side of the cell to the outer side of the cell, and obviously the outer side of the cell contains higher concentration of H positive ions as compared to the inner side of the cell, and this results in the creation of an electrochemical potential gradient, or you can say proton motive force. Okay, this proton motive force stores energy. When H positive ions move back towards the inner side of the cell through this transporter protein, they also drive the movement of some other molecules, say for example glucose molecules, from outer side to the inner side of the cell against the concentration gradient. Here is less concentration of glucose, here is more concentration of glucose. When these H positive ions move back to the inner side of the cell, they also drive the movement of these glucose molecules from their lower concentration to higher concentration. This type of transport process in which the energy stored in electrochemical potential gradients is used to transport some other molecules from their lower concentration to higher concentration. This is known as secondary active transport. Now let us focus today on primary active transport and its types. In primary active transport, the membrane proteins that carry out the transport process, they are known as pumps. You have to remember that those transport proteins that carry out primary active transport, they are known as pumps. Here they are known as pumps, okay? And most of these pumps, they transport ions such as H positive ion or calcium dipositive ion. And further, these pumps are divided into two types. One is electrogenic ion pumps and second one is electroneutral ion pumps. So what are electrogenic ion pumps? Those pumps which involve the net movement of charge across the membrane, they are known as electrogenic ion pumps. Those ion pumps which move ions in such a way that they create net movement of charge, they lead to net movement of charge across the membranes. For example, the first one, the example is here, sodium potassium ATPase. It is an electrogenic pump, okay? The sodium potassium ATPase of animal cells, it pumps 3 sodium ions out of the cell and 2 potassium ions inside the cell, and this results in a net outward movement of 1 positive charge. यहाँ पे आप देख सकते हैं, here this is the sodium potassium ATPase pump. By consuming energy in the form of ATP hydrolysis, it drives the movement of 3 sodium ions out of the cell, and it also drives the movement of 2 potassium ions inside the cell. This leads to the creation of a net charge difference between the inner side and the outer side. Here we have more positive charge on the outer side and less positive charge on the inner side, okay? So those pumps which lead to charge difference between the outer side and the inner side of the cell, they are known as electrogenic pumps. And now the second type is electroneutral pumps. Those pumps which function in a way that there is no net movement of charge across the membrane, they are known as electroneutral pumps. And the example is H positive, K positive ATPase of animal gastric mucosa. It pumps 1 H positive ion out of the cell for every K positive ion which moves inside the cell. So there is no net movement of charge across the membrane. Here you can see this is the H positive, K positive ATPase pump of the animal gastric mucosa, okay? It moves 1 H positive ion outside the cell and 1 K positive ion inside the cell, so that there is no net movement of charge, there is no net separation of charge, or you can say there is no net charge difference between the inside of the cell and the outer side of the cell. This type of pump is known as electroneutral pump, okay? This is all about primary active transport. I hope you all understand this. See you soon with the next lesson. Till then, thank you and